Welcome to our deep dive into the world of veterinary neurology. We're going to be talking about a study that could revolutionize how we diagnose brain disorders in animals.
Yeah, this study published in Frontiers in Veterinary Science is focused on electroencephalography, or EEG, in unsedated dogs and cats.
So you're saying we can get EEG readings without having to use anesthesia?
Exactly. For years, we've relied on anesthesia to get clear EEG readings in animals.
And that can be stressful and risky for our pets.
Absolutely. This new study is a huge leap forward because it eliminates that need for anesthesia in most cases.
That's exciting news for pet owners. So how did these researchers manage to pull off EEGs on awake animals? What's the secret?
Well, they developed a special cap called the PET cap, which holds the electrodes in place.
Okay, so it's like a little helmet for their brainwaves.
You could say that. And along with the PET cap, they use a recording system that captures not only brainwaves, but also video and other physiological data.
So they can see what the animal's doing while they're recording the brain activity.
Exactly. That way they can correlate the behavior with the brain activity.
That makes a lot of sense. So it's all about making the animal comfortable and getting a clear picture of what's happening in their brain.
That's the goal. And the results are really impressive. They conducted 150 EEG recordings on 140 animals.
That's a pretty good sample size.
It is. And out of those 150 recordings, only 9 required sedation.
Wow. So that's like a 94% success rate.
You got it. That's a huge improvement over traditional methods.
Absolutely. So what kind of information did they get from these unsedated recordings? Were they able to see different brain states?
Oh, absolutely. They were able to see wakefulness, drowsiness, and sleep in 99% of dog EEGs.
And what about cats?
91% of cat EEGs showed those different brain states.
So we're talking about clear, readable brainwave patterns without the need for anesthesia.
Exactly. And they also looked at something called readable percentage, which refers to the portion of the recording that's free from artifacts or noise.
So the cleaner the signal, the higher the readable percentage.
Right. And they found that 75% of the dog EEGs and 83% of cat EEGs were readable for more than half of their duration.
Which was how long?
About 40 minutes.
That's pretty impressive. 40 minutes of clean brainwave data without anesthesia. That seems like a really valuable tool for diagnosing neurological problems.
It is. It opens up so many possibilities.
Now, you mentioned that they looked at impedance values. Can you explain what those are for our listeners who might not be familiar with the term?
Sure. Impedance values basically tell us how well the electrodes are picking up the electrical signals from the brain.
So the lower the impedance, the better the signal?
Exactly. And they were able to achieve impedances below 20K in 99% of dog EEGs and 98% of cat EEGs.
Okay, so the signals were strong and clear. But what about making sure the signals were accurate? Did they encounter any issues with things like isosynchrony?
Ah, yes, isosynchrony. That's when nearby electrodes pick up similar signals, making it look like brain activity where there is none.
Kind of like a false positive?
Yeah, exactly. But the good news is that they found isosynchrony only affected a very small percentage of the channels, around 6%.
That's reassuring. It sounds like they took a lot of care to ensure the accuracy of the recordings.
Absolutely. They wanted to make sure the data was reliable.
So we've established that these unsedated EEGs are clear, reliable, and capable of capturing various brain states. But the big question is, how do they actually use this to diagnose neurological problems?
Well, that's where it gets really interesting. In 32% of the animals, the EEG revealed abnormalities that directly helped in making a diagnosis.
So they were able to see something on the EEG that correlated with a neurological issue.
Yes. In some cases, those abnormalities matched up perfectly with the animal symptoms.
To confirm what the vets were already suspecting.
Exactly. But here's a really exciting part. In 7% of the cases, the EEG picked up abnormalities even when the animal wasn't showing any symptoms during the recording.
So they were able to detect potential problems before they even became noticeable.
Precisely. That's the power of unsedated EEG. It can help us catch things early on.
Wow, that's amazing. Talk about early detection. So how do they go about actually provoking brain activity to try to uncover some of these hidden issues? I know you mentioned intermittent photic stimulation, or IPS. What is that exactly?
So IPS involves flashing lights at different frequencies to stimulate the visual cortex.
Kind of like a strobe light effect?
Sort of. It's a technique that's often used in human neurology to help diagnose epilepsy.
Well, they're looking to see if the flashing lights trigger any unusual brain activity.
Exactly. And they found some interesting differences between cats and dogs.
Oh, How so?
Well, a whopping 85% of cats showed a strong response to IPS.
Wow, that's a lot.
It is, but only 11% of dogs reacted to the flashing lights.
That's a pretty big difference. Why do you think that is?
That's the million-dollar question. The study suggests that it might have something to do with the unique structure of the feline visual system.
Interesting. So cats are just more visually stimulated than dogs?
It seems that way, but more research is needed to really understand why there's such a difference in their responses.
Makes sense. It's fascinating how much we still don't know about animal brains.
Absolutely. There's so much more to explore.
So IPS was one way to stimulate the brain. Did they use any other methods?
They did. They also used more tailored stimuli based on each animal's history. For example, if a dog was suspected of having reflex epilepsy triggered by eating, they would offer the dog food during the recording.
So they were basically trying to trigger a seizure in a controlled environment.
Yes, exactly. And in some cases, they did successfully trigger seizures, and they were able to see the corresponding epileptic activity on the EEG.
It's amazing. It's like they were able to capture those seizures in action.
Right, and that can be really helpful in determining what kind of seizures a dog is having and how best to treat them.
It seems like this unsedated EEG could be really game-changing for veterinary neurology.
I think so too. It gives us a whole new way to look at the brain.
Now, I know this study was focused on diagnosing neurological problems, but I'm curious about whether they had a control group of healthy animals who underwent the same protocol.
That's a great question, and the answer is no. They didn't have a control group in this particular study.
So it's hard to say for certain whether some of the EEG abnormalities they saw are actually indicative of a problem or just normal variations.
That's true. More research is needed to establish those baseline parameters for healthy animals.
It would be interesting to see what future studies reveal.
Definitely. But even without a control group, this study provides a lot of valuable information.
Absolutely. So let's talk about the practical implications of this technology. How feasible is it to incorporate unsedated EEG into everyday veterinary practice?
Well, one of the key things is creating a calm and comfortable environment for the animal.
So no more bright lights and scary exam tables.
Right. Ideally, you want to minimize sensory overload, soft lighting, familiar scents, maybe even letting the pet sit in their owner's lap.
That makes sense. A relaxed pet is going to be more cooperative.
Exactly. And the equipment itself is relatively portable. You have the pet cap with the electrodes, the EEG recording device, and a video camera.
It sounds pretty straightforward.
It is. Of course, there's always the question of the conductive gel.
Yeah, I was wondering about that. I can imagine it getting all matted in the animal's fur.
Well, the researchers recommend using a conductive paste like 1020 followed by a gel like Signagel. These products are designed to be easily removed with dry shampoo.
Okay, so no messy fur situations. That's good to know. So once the pet cap is in place and the electrodes are prepped, what happens next?
Then you just start recording. The pet can relax, have some treats, maybe get a little ear scratch while the EEG is capturing their brainwaves.
And the video camera is recording their behavior at the same time.
Exactly. So you can see if there are any behavioral changes that coincide with changes in brain activity.
That's really cool. It's like having a window into their brain.
It is, and it can be especially helpful in diagnosing conditions like epilepsy.
Because you can see exactly when a seizure starts and how it affects the brainwave patterns?
Exactly. And what about its ability to detect those subclinical problems, the ones that aren't even noticeable yet?
That's what I find so fascinating about this technology. It's like we can see beneath the surface.
Exactly. By removing the effects of anesthesia, we get a much clearer picture of what's happening in the brain.
So we might be able to diagnose conditions like epilepsy or brain tumors before the animal even starts showing symptoms.
That's the hope. Early detection is key for so many neurological conditions.
It could really make a difference in their long-term health and well-being.
Absolutely. It's about shifting from reactive care to proactive care.
I love that. So we talked about the benefits of unsedated EEG, but what about the challenges? Are there any hurdles that need to be overcome?
Well, one challenge is simply access to the equipment. The specialized pet caps come in different sizes to fit different head shapes, so not every clinic will have them on hand.
Right, so it's not a one-size-fits-all solution.
And then there's the cost factor.
Yeah.
EEG equipment can be expensive.
I can imagine. But hopefully, as demand grows, we'll see more affordable options become available.
I think that's likely. But another important consideration is expertise. You need someone who knows how to read and interpret EEGs properly.
It's not just about having the equipment. It's about knowing what to do with the data.
Exactly. And you also need someone who is comfortable working with awake animals.
Because not every animal is going to be thrilled about wearing a cap and having electrodes attached to their head.
True. Some animals might need a little extra coaxing or even mild sedation.
So it's not always a completely hands-off approach.
Right. It's important to prioritize the animal's welfare above all else.
That's a good point. So even though unsedated EEG is a promising advancement, it's not a magic bullet.
It's a tool, and like any tool, it needs to be used appropriately.
Well said. So we've talked about the logistics, the challenges, the expertise involved. But let's step back for a moment and look at the bigger picture. What is the long-term potential of unsedated EEG in veterinary neurology?
I believe this technique has the potential to revolutionize the field. Just like EEG transformed human neurology, unsedated EEG could become a standard part of veterinary care.
So we could be talking about routine, non-invasive EEGs for all sorts of things.
Exactly. Not just for diagnosing diseases, but for monitoring treatment progress, assessing brain health in older animals, and even for studying animal behavior and cognition.
It's about understanding our animal companions on a much deeper level.
Yes, and using that understanding to provide them with the best possible care.
That's a beautiful thought. It sounds like there are some really exciting possibilities on the horizon for unsedated EEG. What else can we look forward to?
Well, one area that I find particularly exciting is the study of animal cognition.
Oh, yeah, you mentioned that earlier. What do you mean by that?
Well, imagine using unsedated EEG to get a glimpse into how animals perceive the world, how they learn, how they solve problems, maybe even how they experience emotions.
Wow. So we could actually start to understand what it's like to be a dog or a cat.
Exactly. We could study how they process language, how they navigate their environment, all sorts of things.
That's incredible. What about animal emotions? Can we really measure something like that with EEG?
That's a really interesting question, and it's a bit of a debate in the scientific community.
Because emotions are so subjective.
Right. But even though we can't directly measure emotions like happiness or sadness, we can detect changes in brain activity that are associated with different emotional states.
So we might be able to see patterns that correspond to things like fear or anxiety.
Exactly. For example, researchers have already used EEG to study anxiety in dogs.
Oh, but what did they find?
They were able to identify specific brainwave patterns that are associated with fear and stress.
Wow, that's amazing. So this technology could help us understand and treat emotional disorders in animals.
Absolutely. It's about taking a more holistic approach to veterinary medicine.
Treating the whole animal, not just their physical symptoms.
Exactly. Unsedated EEG could be a really powerful tool for that.
It sounds like it. It's really exciting to think about all the possibilities.
It is, and it's just the beginning.
I'm sure we'll be hearing a lot more about unsedated EEG in the years to come.
I think so too. It has the potential to transform the way we care for animals.
Well, on that note, I think it's time to wrap up this deep dive.
It's been a pleasure talking with you.
Likewise. For our listeners out there, if you're interested in learning more about unsedated EEG, I encourage you to talk to your veterinarian.
They can tell you if it's an option for your pet.
And in the meantime, from Vet NeuroJar, keep those minds inspired, hearts light, and tails wagging.